This is the first half of chapter 6, so we call it 6a, referring to bones in the skeletal tissue. You have to understand how the bone is made up, the structurally, so the anatomy of it, and how it's continuing throughout your lifetime. Certain remodeling processes, depending on the stresses that are placed on it. You need to know this as a healthcare worker so that you know how to deal with the patients when there has been an injury or some type of disease or disorder that affects the bones. Obviously, you have to know how things occur under normal conditions so that you know how to deal with them now that there's some abnormal condition. So the human skeleton initially, when it's first formed, in terms of embryonic development, is just cartilage. Now, the cartilage is going to be replaced by the bone, which is known as ossification. Cartilage will remain in certain areas where you have much more flexibility. So skeletal cartilage, when we look at it, it's very highly resilient. It's composed mostly of water. One thing to keep in mind is that there are no blood vessels, excuse me, or nerves in skeletal cartilage. The perichondrium, this is a layer of dense connective tissue that is surrounding the outside of the cartilage, kind of holding it all together. It helps the cartilage, when it grows, to grow in a specific direction. And it keeps it from just visualized like a blob just growing outward. It keeps it contained. The perichondrium does have blood vessels in it, so that by diffusion, those nutrients, that oxygen, etc., can be supplied to the cartilage, since the cartilage doesn't have its own blood supply. Cartilage is made up of chondrocytes. These are cells that are encased in little cavities that we call the lacunae. And there's an extracellular matrix that's surrounding it. Excuse me. There are three types of cartilage. There's hyaline cartilage, elastic cartilage, and fibrocartilage. Hyaline cartilage does provide a lot of flexibility, a lot of support, resilience. It is the most common type. It has only collagen fibers in it. Some examples would be your articular cartilage, articulations or joints. Costal cartilage, anytime you see the word costal, think ribs, so that's in your ribs. With the respiratory and the nasal cartilage, so the tip of your nose is hyaline cartilage. Elastic cartilage is very similar, but it does contain elastic fibers instead of collagen fibers. Some examples of this would be your epiglottis and your external ear. Fibrocartilage contains very thick collagen fibers, gives a lot of strength. Some examples would be in your knee joint, you have menisci, helps to work kind of like a shock absorber. And then also the vertebral disc. That's the cartilage that's between each of your vertebrae. And so in this diagram, it is showing with the skeleton, number one, it's color-coded for the bones of the skeleton by whether it's the appendicular skeleton, which are the appendages and how it attaches, to the axial skeleton, which is the torso, the head, the neck, along this area. Now you also have color coding for the three different types of cartilage. So you have got the color coding for the blue is the hyaline cartilage. So here you've got your costal cartilage, your articular cartilage and joints, elastic cartilage, remember, like in the external ear. And then fibrocartilage is a little bit hard to see, but it's right here, is between each of the vertebrae as an example. So cartilage can grow in two ways. It can grow by appositional growth or interstitial growth. Appositional growth is when the cartilage basically is going to be growing, and we see this with bone growth as well. So appositional is when you're on the exterior surface and you're laying down new matrix then on the surface. So it's like you're growing outward in diameter. Interstitial growth, what's going to happen here is those chondrocytes that are in those little lacunae, those little, they look like little open vessels, they will divide and they're going to secrete new matrix as connective tissue does. Cartilage, remember, is connective tissue, so it has that matrix. So now you're growing essentially from within. So if you're growing on the external surface, appositional growth. If you're growing, think of it more internally and spreading out, then that's interstitial. Calcification of cartilage does occur during normal bone growth and use. It can occur later in life in certain situations. Calcification and hardened cartilage is not the same thing. Okay, so hardened cartilage is not the same as bone. So what are the functions of the bone? Functions of your skeletal system. Number one is for support. It does help to support a lot of the organs, hold them in proper places, like the bones of the pelvis are helping to hold a lot of your abdominal and pelvic organs. For protection, if you notice, your most vital organs, such as your spinal cord, your brain, your heart and lungs, they are surrounded by bone. Movement. They provide an attachment point for your muscles so that when the muscles contract, they're going to be moving the bones. And that, whether you're walking, moving your arm, etc., it's because the muscles are attached to the bone. Bone's also important for both mineral and growth factor storage. Things like calcium, when there's excess of it in the blood, it gets stored in the bone matrix. It's important for blood cell formation. Hematopoiesis is the production of new blood cells, especially red blood cells. So it's occurring in the red marrow, which is found in the cavities, the medullary cavities of certain bones, especially like your long bones. It's important for storage of fats like triglycerides. Now, you might say, why are you storing fats in bone? Well, it can be stored also in those medullary cavities in the yellow marrow as an energy source. Usually the body's going to use carbohydrate for energy, but if it runs out of that, then it can go to the fat and break that down for energy. And then there's also another function of bones, which is hormone production. A lot of people don't necessarily think about it. The osteocalcin is secreted by bones. It's going to help with regulating insulin secretion, which is involved with glucose levels, which is related to your metabolism. So there's several different functions of the bone. How many bones are there? There are at least 206 bones. Now I say at least. Some people have more than that. In the sutures, which are the joints of the skull, where they come together, they're immovable joints. It's kind of like piecing together. It's not a straight line to a straight line. They tend to have jagged edges. It's kind of like putting together puzzle pieces. And sometimes there are small little, what we call sutural bones in there. And that number varies from individual to individual. But the number of bones that are the same between each other is 206. You will be learning the names of the bones. We are focusing on this chapter on how the bones are made. Naming of it, we tend to focus more on the actual naming in the lab portion. As I mentioned just a little bit ago on the previous slide, the skeleton is divided into two groups. We love to subdivide things, and it's based on its location. So the axial skeleton, as I said earlier, that's going to be the skull, your rib cage, your vertebral column, kind of along that torso. The appendicular skeleton essentially are your appendages, your arms, your legs, and then the attachment points. So once again, that same picture from before. This darker kind of brownish tan color is the axial skeleton, and the light tan color is your appendicular. Now, we can also classify bones not only by their location, but we can classify them by their shape. You will notice, if you haven't figured out yet, that in biology we often have multiple ways of classifying things. So the bones can be classified into 4 basic shapes. You have long bones. The name implies they're longer than they are wide. This is typically found in your arms and your legs. Short bones, found in like the wrist and the ankle. Now, some books will consider a 5th group by itself, which is the sesamoid bones. This book puts it as like a subgroup of the short bones. Sesamoid bones are always going to be found within tendons. And the classic example is your patella, which is your kneecap. Flat bones, they're very thin, they're flat, they may be slightly curved, often found in the skull, so a lot of the ones in the skull are slightly curved. Your sternum. Irregular bones, these are the ones that, frankly, they don't fit in any other category. It's like, oh, what kind of shape is this? Well, your guess is as good as mine. It's an odd shape. Things like your vertebrae, they just don't fit in any of those other categories. And so as you can see on this diagram, I mean, a long bone, like I said, usually on your appendages. Here's a flat bone, short bone, like I say, your wrist, your ankles. But the vertebrae, see how it doesn't really fit in any of the other categories. Bones are classified as an organ. Remember, an organ has at least 2 different tissue types that works together to carry out a function. Well, bones, you might be thinking, well, wait a minute. It has osseous tissue or bone tissue, which is a type of connective tissue. But what other type of tissue does it have? Well, it does have nervous tissue. It has blood vessels in it, which has the blood flowing through it. So because it has more than one type of tissue, it is classified as an organ. It may have more than just the bone tissue. It may have like fibrous connective tissue. It may have muscle cells with it. It may have cartilage on it. We're going to look at the 3 different levels of structure: the gross anatomy, microscopic, and then the chemical. So we look at the gross anatomy. You have compact and spongy bone. Compact bone is always going to be on the outermost layer. It's going to look very smooth and very solid. It's very dense. It's very strong. Spongy bone is always going to be in the interior. And it gets its name because it looks like a sponge. It has all these little openings, these little small flat pieces of bone interconnected like scaffolding that are called the trabecular. The open spaces between all these tuberculi, like the open spaces of the sponge, these are usually filled either with red marrow or yellow marrow. Now, some of your short, irregular, and flat bones usually will have the compact bone on the outside and then a thin layer of spongy bone on the inside. It kind of looks like a sandwich, if you will. Periosteum covers the outside of the compact bone, and the endosteum covers the inside portion of the compact bone. Then, like I said, that bone marrow is going to be within the spongy bone. Hyaline cartilage covers areas of the bone if it's part of a movable joint. So this is showing on the bottom picture the trabeculi. That's what the bone arrows are pointing out here, these little bone projections. These spaces, as I said, are going to be filled with either red marrow or yellow bone marrow. And it looks like it's just kind of randomly placed through here. They've actually had structural engineers look at spongy bone and the orientation of all of these tuberculi, and they have found from a structural standpoint, from an engineering standpoint, it's kind of like, hey, this is a pretty good job. This is providing a lot of strength. Now, if you notice here, like I said, this is found in the flat bones, the short bones, the irregular bones. You have the compact bone on the outside, which makes sense because you don't want this directly exposed for danger of breaking it. So the spongy bone is always going to be internal on the inside layers, and that's why I said it kind of looks like a sandwich, if you will. The spongy bone is, as you can see, it's much less dense than the compact bone. That makes this bone, the entire bone overall, much lighter. If this were solid compact bone, it would make the skull much heavier, and you'd have problems holding your head up. Now, when you look at the structure of a long bone, it's a little bit different. All the long bones are going to have a shaft, what you commonly refer to as that shaft in the middle. We call that the diaphysis. The ends are known as the epiphyses, and then there's going to be membranes associated with it as well. The diaphysis, I said, that's commonly referred to as the shaft. It forms that long axis of the bone. Now, internally, you've got the compact bone outside, but internally, you're going to have what's known as your central medullary cavity. That will be filled with yellow marrow in adults. Children, it's usually initially filled with red marrow. On the ends, you've got the epiphyse. Now, internal, you've got, once again, spongy bone, external, compact bone. If it becomes, and most of your long bones are part of a joint, the ends, they form a joint with an adjacent bone. So there's going to be articular cartilage on that surface that's involved with the joint, not the entire bone, just on the end. Between the diaphysis and the epiphyse, where they come together, is what's known as the epiphyseal plate or epiphyseal line. In a child who is still growing, it's referred to as epiphyseal plate, and that is where you actively have bone ossification occurring, growth of bone. In an adult, once you have reached your full height and you have stopped growing in height, then it's referred to as the epiphyseal line, and you can detect this on an X-ray. So if we look at a representative drawing of a long bone, I said the diaphysis is that shaft. So you've got the compact bone, always, no matter where you're at on the bone. Compact bone is going to be on the outside. Spongy bone internally. Now on the diaphysis, at the very end, you're going to have spongy bone, but in the middle, the majority of this is going to be your medullary cavity, and with an adult, like I said, it's going to be filled with yellow bone marrow. The periosteum is this membrane that's on the outer surface of the bone. It kind of becomes continuous with the articular cartilage on the end that's involved with the joint. The endosteum is lining the inside. It's not shown here right now, but it's on the inside. The epiphyse will be the end. Because you're talking about long bones on your appendages, you're going to have a proximal end and a distal end. Once again, your articular cartilage, your spongy bone internally. Right here is the epiphyseal line. Like I said, this would be the epiphyseal plate in a child. This would be where the growth would be occurring in this direction in a child. So if we look up a little bit closer at this, once again, the spongy bone, the compact bone on the external surface, and then this is taken from the epiphyse, so it's the articular cartilage here. And then notice on the lining on the between the compact bone and the spongy bone, that's where your endosteum is. Think endo-internal. And notice that, yes, bone is actively growing. Even in an adult, it's actively remodeling, depending on particular stresses. So it needs to have a very good blood supply. So you'll notice that you have blood vessels going through it. So the membranes, the periosteum, it said it covers the external surface. There's fibrous layer, that's the outer layer. It's very condensed, irregular connective tissue. It contains what are known as Sharpey's fibers. That helps to hold it to the bone itself, to that bone matrix. The osteogenic layer is the inner layer right against the bone, and it does contain osteogenic stem cells, which is the precursor to most of your bone cells. It's going to have a lot of nerve fibers and blood vessels. that will run through what are known as nutrient-forming openings to bring that blood supply and nerve to the internal portion of the bone. The periosteum is the anchoring spot. That's what a lot of your tendons and ligaments are going to attach to and once again kind of become continuous with. Then the osteum is the membrane on the internal surface of that compact bone. It lines all the canals that pass through the compact bone. It can also contain osteogenic cells, which once again are the precursors to all the other bone cells. So once again, from another diagram here, internally you see the endosteum, you see the compact bone, the medullary cavities filled with that yellow bone marrow, the periosteum on the external surface. And these fibers here, those are the Sharpey's fibers that are connecting and holding the periosteum to the bone. You don't want this membrane, periosteum membrane, just kind of sliding up and down. It needs to be held tightly in place. And then once again, your blood vessels supplying the needs of the internal portion of that organ. Hematopoietic tissue in the bones, the red marrow is found within trabecular cavities of the spongy bone. In newborns, medullary cavities and all spongy bone contains red marrow. In adults, the red marrow is going to be found in the head portion of the femur and the humerus. And then most active areas for the hematopoietic, which is the formation of blood cells, occurs in the flat bone and some of your irregular bones, like your hip. Yellow marrow can be converted to red if a person becomes anemic. Bone markings, these are places or sites where you can have muscle attachment, ligament attachment, tendons attachment, and some of the markings are usually involved with joint formations. They may be openings for blood vessels and nerves. Blood vessels and nerves often run together, and so there may be markings of where those are lying as they're adjacent to the bone and then how they enter the bone. So 3 types of markings: projections, which is an outward bulge of the bone. It's often a modification for joints or where you have a muscle attached and there's increased stress from that muscle pull. Depressions are kind of like a bowl or a cutout that can serve as passageways, like I said, for the nerves, the blood vessels, or also playing a role in the joint in the way adjacent bones have to fit together. Openings, as the name implies, it's a hole that serves as a passageway for the blood vessels and the nerves. You can get some indications if you find a skeleton as to kind of the level of activity that that individual had by the bone markings. The more pronounced the bone markings are, the more stress, etc., that they probably had, say, on projections. Somebody who was very active athletically would tend to have more pronounced bone markings than, say, someone who was very sedentary. So you can tell an awful lot from a skeleton. As they say, a dead man tells no lies. He can tell you from a skeleton how active the individual was. We can tell whether the person was a child, not only by size, but by the epiphyseal plate versus epiphyseal line. Had they finished growing? Young infants, we can tell if you have the skull, basically if they were under the age of 2. Oftentimes, depending on which bones you find, you can determine whether it's a male or a female. So there's quite a bit of information that can be gained just by looking at the skeleton. And this goes through the tables in your book, the various type of bone markings. For my classes, I tend to focus on this more in the lab portion. We tend to separate from my class The lab tends, certainly in A&P1, tends to be more focused on anatomy, and in the lecture we focus more on the physiology. But just to kind of briefly go through things like tuberosities, all of these are projections. So you have various tuberosity, crest. So this would be a site for muscle attachments. It's usually fairly prominent. You have different processes, which is any type of a bony projection out. Spine usually is a very sharp and slender, oftentimes a pointed projection out. A head, a facet around us. Often these times these are helping to form joints. So it's going to be how they connect together to form a joint. And then the depressions and openings. The foramen magnum is opening at the base of the skull that allows the spinal cord to come up and connect directly to the brain. You do have foramens in several of your facial bones that allow for blood vessels to move from the external surface internally, etc. One of the things, if you haven't done so already, to help you study, certainly I find with the skeletal system and learning the names of the bones just in general, but then also these projections, is to print off pictures. You can find a ton of pictures. Just go to Google Images and just type in whatever you're studying at that point. Print off a copy and then slide it into a plastic sleeve that you can then write on the plastic sleeve, naming it. And to practice with that, then you can write it with a marker, erasable marker. Don't use a Sharpie, use an erasable marker. And then you can wipe it clean, and you can do that over and over, and that repetition really helps with learning. What I often recommend, and this is just my own personal recommendation, you can take it or leave it, is that I would recommend that first learn the bones and then go back and learn the markings, because to me it just seems to make more sense. Number one, it's not going to do you any good to know the markings on a bone if you don't know what the bone is and you don't know where that bone is. If you don't know where the tibia is, great that you know all the markings on it, but do you know where the tibia is? Do you know what its main purpose is? That's not going to help you a whole lot. So if you look first at learning the names of all the bones, then once you know where the bone is, you know the proper name of it, then go back and start learning some of these bone markings. And it's going to make a little bit more sense of, oh, okay, well, this does make sense that the tibia is transferring the weight of the body from the upper body, from everything above, from the femur basically, down to the foot. So that means it has to be connected to the femur, but it also has to be connected to the ankle, which are the tarsal bones. So now I've got myself in the right location. Well, there's going to be also a lot of muscle attachment there. But if you compare the tibia, say, to the fibula is much smaller. Why? Because it's not transferring weight. It's more for muscle attachment. It's the tibia that's transferring the weight down. So it must be bigger. It must be able to handle the weight. The fibula is not weight bearing. And it's going to help you to realize, once again, okay, if I'm part of a joint, let's see what the end of this joint looks like. Okay, so this makes sense. Learn the bone first and then go back and learn the markings. Now, if we move on to the microscopic anatomy of the bone and we look at the cells that are found in bone tissue, there's 5 major types of cells. There are the osteogenic cells, the osteoblasts, the osteocytes, the osteoclasts, and the bone lining cells. And I know it's really annoying because they all sound alike. Obviously, osteo refers to bone. So at least that's a clue that's going to help. The osteogenic cells, these sometimes are called the osteoprogenitor cells. They're very active going through mitosis. Remember, I've already mentioned those just a few slides back when we were talking about the periosteum and the endosteum, saying that you have these osteogenic stem cells. They are the precursors to all of the other cells. So here's what's going to happen is that they are stimulated to go through mitosis, through cell division. And one of them will always remain as an osteogenic stem cell, so that can continue going through mitosis. Then they will differentiate. One of them will differentiate into either an osteoblast or a bone lining cell. So what are the osteoblasts? Think bone forming. These are the cells that are going to be secreting, making the bone matrix. Now, before it mineralizes, this bone matrix is called the osteoid. It's made up of collagen, it's made up of calcium binding proteins in there. So initially, these osteoblasts are very active mitotically. So here's your osteogenic cell, it's a stem cell, and it differentiates into the osteoblast. Think bone forming. The osteocytes, these are mature bone cells that are in those lacunae, but they can no longer divide. They help to maintain the bone matrix. They help to detect stress or any strain. Think of them as like a sensor. They're within the bone matrix, and they're going to be the sensor. They're going to respond to different stimuli, whether it's increased force, whether it's weightlessness, and they are going to take that information. So it's kind of like I'm a sensor and I'm here. I can no longer divide because I'm stuck in the lacunae, but I can communicate to osteoblasts and osteoclasts. Now, we just talked about the osteoblasts that are forming bone matrix. Osteoclasts are destroying the bone matrix. So they're the opposite of the blast. And then we also have the bone lining cells. These are cells that are on the surface of the bones. They help to maintain the matrix. On an external bone surface, these can also be called the periosteal cells. On internal, endosteal cells. And then the osteoclasts. They basically are involved with bone resorption, breaking down the bone matrix. And here's a picture of them. And then here's the osteocytes. So what happens is you have your osteogenic cells, which essentially are your stem cells. They will differentiate or specialize and become osteoblasts, which are secreting the bone matrix around them. As bone is forming in this bone matrix, which is called the osteoid, when it's initially made, within a few days it will harden. And some of those osteoblasts now are stuck within these little, like, open, they look like little cavities. That's the lacunae. And once an osteoblast is stuck in there and it can no longer divide, it becomes the osteocyte. It has long extensions, so it can communicate with osteoblast and osteoclast. When it detects stress, when it detects any type of information, then it can send, and it gets a stimulus, and it can, oh, okay, we need to build up more bone. It will stimulate the osteoblasts that are throughout the bone to secrete more matrix. We need to make more bone. If the reverse is true, where it gets a stimulus, no, we need to start breaking down some of the bone matrix, then it stimulates the osteoclast to say, okay, you need to start breaking down the bone matrix. Just to give you an idea, one of the stimuli that it may be receiving may be, what is the concentration of calcium in your blood? That's a huge thing that is involved with this balance between the osteoclast and the osteoblast, because excess calcium is stored in the bone matrix. So if calcium levels get too high in the blood, then it's going to stimulate the osteoblast. Put some of this calcium, start building bone matrix up, because that's where we need to store the extra calcium. If calcium levels get too low in the blood, then it's going to do the reverse and stimulate instead the osteoclast to say, you need to start breaking down some of that bone matrix, because we need to get that stored calcium that's there, we need to get it into the blood, get those levels up in the blood. So osteogenic, think genesis, it starts the whole process, it's a stem cell, it becomes osteoblast. Some of the osteoblasts become your osteocytes, which receive the stimulus. They will then send the mess. Think of them as the messenger. Osteoblast bone-forming. Osteoclast is breakdown of the bone. Compact bone is going to be consisting of several different things. Your osteon, which is your Haversian system, different canals and little smaller ones that are called the caniculi, and interstitial and circumventral lamina. So the osteon, which is also known as the Haversian system, the osteon is the structural unit of compact bone. So what does it look like? It looks like this real long cylinder that's going to run parallel to the long axis of the bone. They act like weight-bearing pillars, weight-bearing columns. Just like you have weight-bearing columns in a building or in a home, it's the weight-bearing pillars. It's composed of several rings of bone matrix that's called the lamellae. These have collagen fibers. In adjacent rings, the collagen fibers are going to be going in different directions, because this adds additional strength. It helps to prevent twisting. And then you've got bone salts that are going to be found between the collagen fibers. The collagen fibers do provide, I said, some flexibility. The bone salts are going to provide a lot of hardness and strength. A very easy experiment if you want to do at home, especially if your kids, they might enjoy this. You can take, say you're cooking chicken. Next time you do that, you're baking chicken, and everybody knows when you cook the chicken afterwards, the bone, you can break. If you were to push against it, it's going to break. When you bake, let's say it's chicken, when you bake it, the high temperature destroys the collagen fibers. So the only thing left would be the salts of the bone. So you've lost any flexibility that might have been there from the collagen fibers, because they were destroyed by the heat. What you can do is take, before you cook the chicken, take a bone. It works best like if you use the leg bone. But take the bone, pull all the meat off, pull all the cartilage, clean it as best you can before you cook it. Put it into a container. When I've done this, I usually put it in like a Tupperware container, something that you can seal. Add vinegar to it, enough so that the bone is completely covered with the vinegar. And then I say seal it. You want to seal it because you need to let it sit. Sometimes I've let it sit for as long as 2 weeks. Usually a week is good, but sometimes 2 weeks has worked well. And so that's why you want it covered, because remember, this is the raw chicken leg with the meat pulled off. After you've let it sit for about 2 weeks. What happens is the vinegar is destroying the bone salts so that all you're left with is the collagen fibers. And you can take that bone, wear gloves. Sometimes what I do is I'll take it out and I put it into, say, a sandwich bag that you, like a Ziploc, something where you can seal it. And because the bone salts have been destroyed by the vinegar, like I said, all you're left with are the collagen fibers, which gives that flexibility to the bone. Not obviously a whole lot, but you want to have a little bit of give and take. Well, when you've destroyed all the salts, that bone, after about 2 weeks in vinegar, it's going to kind of look like a rubber band that you can bend it. And the best way to describe it is it's the flexibility of it. It's kind of like a rubber band. So I say, you might enjoy doing this. If you have kids, they might get a kick out of seeing it. But it really brings to light the two properties that the bone salts bring: that hardness, that strength. The collagen fibers bring some flexibility. It's able to withstand a lot of stress and resist twisting. And you put the two together, and what do you have? You have a very strong bone that can take a little bit of twisting. It can take some stress to it. It's very, very strong. With the lamellae, these are the concentric circles that they look like when you're looking at a cross section. And notice here that the collagen fibers, I mentioned a little bit ago, how they're going in from one layer to another in different directions. So this makes it much, much stronger. And then in the very middle, we have what's known as the central canal, and that's where you have your blood vessels and you have your nerves. That central canal, they used to call them the Hyaline canal, so it's going to kind of going through and changing names, so you may see it as either one. That's going right through that middle. Perforating or Boltzmann's canals, these are going to be coming in at right angles to that central canal. Because how do they get to the central canal? Well, it's going to be through the perforating canals. That's what's going to connect those blood vessels and nerves, the central canal to the medullary cavity and the periosteum. Looking at the small cavities that contain those osteocytes, because remember they were originally osteoblasts. They secreted and made the matrix around them. So basically, yes, it's good they're making bone matrix, but now they're stuck within these cavities. Caniculi are little tiny canals that will connect the loci to each other, and they will connect it to the central canal. Because those osteocytes, they may not be going through mitosis, but they are still living, which means they still need blood supply. They still need oxygen. They still need nutrients. And so they need to be connected. When that matrix hardens, the cells, like I say, they become trapped in there. But the caniculi allows for the communication still between them. Interstitial lamina are not part of the osteon. They have to fill gaps between the osteons. Circumferential lamellae, these are running just deep to the periosteum. They help to resist or prevent twisting. So I think it's easiest when you can see here on this diagram. Here's an osteon, here's an osteon, here's another one. So you have several of these, looking at the cross-section of the long bone, that are surrounding. In the middle is your spongy bone with the medullary cavity. These osteons are found only with the compact bone. They're not with the spongy bone. You have your central canal. As I said, it's running along the long axis of the bone. The perforating, they're at that 90-degree angle, because remember, your blood vessels, your nerves have to get from the outside into the bone. So down here would have been a perforating canal connecting here. Now it's connected to this one. Now here. It's kind of like you have to have your exits off your highway type things to connect everyone together. When you look at, well, first, here's your osteum. Remember, you've got different layers. The collagen fibers are running in different directions here, making it really, really strong. Your circumventral lamellae are surrounding out here. Once again, if you look at just one of the osteons, here's your central canal. So if you've got these layers, here's your osteocyte. This is the lacunae. There's that cavity because there it was an osteoblast. It secreted this matrix around it, so it became entrapped in this cavity of called the lacuna. How does it connect and communicate to other cells? By these little tiny canicula, little tiny hair-like canals. They connect osteocytes to each other and to the osteoblast. Under a microscope, once again, these little dark areas, those are containing the osteocytes. Remember, the osteocytes are picking up the stimuli of stress, etc., and are stimulating then the osteoblasts to form bone and the osteoclasts to break bone down. That spongy bone, remember, it looks like it's just kind of randomly thrown in there, but it is very carefully organized to help the bone resist any type of stress. I said there are not osteons present, but the trabeculi do contain osteocytes in them. So there are some similarities. You do have a lot of capillaries that are supplying the nutrients. So once again, this is, if you want to refer to the scaffolding, the trabeculi, it's indeed, it is not randomly organized. You tend to find them along stress points, stress lines. So the bone is made up of both organic and inorganic components. This makes up part of the chemical composition of the bone. The organic components, this is going to include all the cells, various cells that we've talked about, the osteogenic, the osteoblasts, the osteocytes, bone-lining osteoclast, and that osteoid. The osteoid is that matrix that's being secreted by the osteoblasts. What's in that matrix? The ground substance, the collagen fibers, that's all helping to give the strength and flexibility to the bone. If there is some breakage that may occur, it may help to, with the collagen fibers, they kind of stretch to help dissipate that energy. They're trying to help prevent fractures of the bone itself, and then some of these bones will reform. The mineral salts make up about 65% of the bone by mass. It's usually, majority of it is calcium phosphate crystals. And as I said, that gives the hardness to the bone. So bone is very, very strong. As you know, it lasts long after death occurs. Part of that has to do because of the minerals. And as I said earlier, it can give us a lot of information when you find a skeleton. Studying ancient people, trying to find out, were they more hunters or more agricultural? When we study the skeletons, like I said, that can tell us a lot of information about it.